Let's think about the human genome as the book of life. For a while now, scientists have gotten pretty good at correcting little typos in that book. But what if we could do more? What if we could insert entirely new, healthy chapters? Well, that's what we're diving into today, a technology that could literally rewrite the future of medicine.
So let's really frame this challenge, right? We've gotten good at fixing those tiny typos in our DNA. But what happens when the problem is bigger? Say a whole page is missing or a section is just completely corrupted. How do you safely insert a huge, healthy piece of genetic code exactly where it needs to go? For years, that's been the holy grail of genetic medicine.
You know, fixing a typo is kind of like a simple search and replace on your computer. It's super useful, but it definitely has its limits. Inserting a whole chapter, though, that means adding a complete functioning gene to replace one that's broken. And that is the next frontier. It could let us tackle complex genetic diseases in a way we've honestly only dreamed about. But this is called a grand challenge for a reason. I mean, trying to insert thousands of DNA letters into the 3 billion letter human genome without causing total chaos, it's been a massive roadblock. If you get it wrong, you risk creating way more problems than you solve. Think of our current gene editing tools like a delivery service. Some of them are just really slow and inefficient. Others can only carry these tiny packages, not the massive genes we actually need to deliver. And worst of all, a lot of them just get the address wrong, dropping the package in a totally random part of the genome, which can have really dangerous consequences. So scientists went looking for a better tool, and they found one in nature. Let me introduce you to the hero of our story today, a family of enzymes called large serine recombinases, or LSRs. You can basically think of these as nature's own heavy-duty DNA delivery trucks, built to cut and paste huge genetic packages. What's so amazing about these LSRs is that they are totally self-sufficient. They don't need to borrow the cell's complicated repair machinery, which just makes the whole process a lot cleaner. After looking around for the best possible candidate, researchers landed on this one specific LSR called DN29. It had a ton of potential, but it was also seriously flawed. And here is just how flawed it was. In its natural state, DN29 only hit the main target site, a spot called at H1, just 12% of the time. Just think about that for a second. The other 88% of the time, it was delivering its precious cargo to the wrong address. You would never ever use a delivery service that unreliable, especially not with your own genes. So the mission became crystal clear. Take this powerful but clumsy enzyme and re-engineer it. They had to turn it into a high-precision tool. And to do this, the researchers put it through this absolutely grueling multi-stage engineering gauntlet. Their whole upgrade process had 3 key stages. First up, directed evolution. Think of it like a genetic Olympics, where millions of versions of the enzyme compete and only the absolute best survive. Second, they started stacking the winning mutations, combining the best traits to create new super enzymes. And finally, they brought in AI to predict the most powerful combinations, blending brute force with really intelligent design. But even after all of that incredible engineering, there was one final crucial addition. The researchers took their new superenzyme and fused it with a guidance system based on the famous CRISPR technology. And this was the absolute final piece of the puzzle. They basically gave their superpowered delivery truck a perfect programmable GPS. This guide, which is a modified version of CRISPR's Cas9 called dCas9, doesn't actually cut the DNA. It just finds the exact address in the genome and acts like a big flashing beacon, telling the LSR, deliver the package right here. So the result of all this work? Well, it comes down to this number, 97%. That's 97% genome-wide specificity. It means that out of all the insertions happening across the entire genome, 97% of them landed exactly where they were supposed to. I mean, going from 12% to 97%, that is just an incredible leap in precision. Just look at this transformation. It's night and day. They took an enzyme that was flat out wrong 88% of the time and turned it into a molecular machine that's almost perfectly on target. This is what turns a research curiosity into a real potential therapeutic tool. So with this incredibly powerful and precise new tool in hand, the next step was obvious. They had to see what it could actually do. Could it perform the kind of heavy lifting needed for real-world medicine? And the results were stunning. This system successfully inserted a massive 12,000-letter piece of DNA. It worked in human stem cells, the very building blocks for future cures. And incredibly, they used it to engineer primary T cells, the exact same cells used in CAR T cancer therapies. All of this with an insertion efficiency that, in this really challenging field, reached an impressive 53% in human cells.
And we're not just talking about easy-to-work-with lab cells either. As this chart shows, they got these impressive efficiency rates in therapeutically critical cells, like stem cells and T cells, using different delivery methods. I mean, these cells are notoriously hard to edit, so getting rates like this is a huge step toward actually using this in the clinic. This quote from the researchers themselves really captures the ultimate vision here. For diseases like cystic fibrosis, where there are hundreds of different mutations that can cause the illness, you wouldn't have to design a unique fix for every single patient. Instead, you could just insert one complete, healthy copy of the gene for everyone, a true one-size-fits-all therapy. The development of this tool is really a landmark moment. It moves us from just, editing the text of our genome to being able to write entirely new, life-saving chapters. And it leaves us with a pretty thrilling question. With this new power, what stories of healing are we going to be able to tell?